Good morning everyone and welcome to everyone who's here in person and online. My name is Deepika. I work at the archives at NCBS and this talk is hosted by the archives at NCBS in collaboration with the workshop for college teachers on using Drosophila melanogaster for biology lab classes. And not that Professor Lakhotia needs an introduction, but I will do a very brief one as he is requested. Professor Subhash Chandra Lakhotia is a cytogeneticist, an academic distinguished professor of zoology, and the Science and Engineering Research Board Distinguished Fellow at BHU. He is known for his pioneering research on Drosophila with regard to its chromosome organization and replication. He contributed to the establishment of the Department of Molecular and Human Genetics at BHU, and he's been associated with several government departments and councils. He's worked with journals such as Current Science and Journal of Biosciences. And he's the recipient of several awards, including the Shanti Swarup Prize. Thank you very much, Professor Lakhotia. And I hand over to you.
Okay, thanks Deepika for the brief introduction, and thanks to Venkat, Deepti and Shashi for organizing this talk, which, as she said, what brings me here right now is the one-week workshop that we are, I'm audible all right? For college teachers to use Drosophila as classroom material. Because what we realized was that students don't have any experimental experience in classrooms, BSc, MSc and so on. So we thought we can introduce Drosophila, and that's something that we have undertaken. We had already one workshop at BHU, and then so for next 5 years we plan to train about, let me see, 150 teachers in different parts of the country so that they can go on to teach more, and that's something that brings me here and take this opportunity to talk about historical, partly because it's archival activity. I thought it would be good to give an idea to young generation what all things had happened about which we often do not know. So my first slide is why we need to know history. And this I quote from a remarkable thing that happened, that Theodor Boveri wrote a classical monograph, which was of course in German in 1914, concerning the origin of malignant tumors. And this was translated in English by Henry Harrison, and that's where he wrote. But see, what is important is most oncologists do not know that Boveri had written this. Partly it's in German, but partly because it's historical. But when one reads it, one realizes that every question that we ask in cancer biology today have been raised there. And he has raised possibilities like chromosome instability was one of the main things that Boveri had talked about. And that's where Henry Harris translated this, and in his own preface, this is what he writes. Regrettably, it can hardly be denied that you can become a competent scientist without giving a thought to the history of your chosen subject, but the history, they're all the same. And if you have only the foggiest idea about what was done before you entered the field, you will have no way of assessing the significance of your own work, and you will certainly overestimate its originality and its importance. Worse still, you may occasionally find that your bright idea was someone else's half a century ago, and that arguments in which you are currently engaged were raging long before you were born, and sometimes with greater acuity. That can be an embarrassment when someone else draws the fact to your attention. And this is important. This is the preamble that I chose, that why we need to talk about things that have gone by, how things develop. And in the same context, what it's also important to know is role of error or misconception in science. Partly because when we teach the process of science, we hardly ever talk about error or misconception that had happened. Only things that succeeded, we know about it. Only those who got it right are honored, others forgotten. But why were they wrong? Why things changed? This is something that we need to know. And some honored scientists whom we all regard very high, and what kind of misconception or wrong thing they did. Einstein, we all know about, Newton, the motion and force and so on. But as the history says, he wrote more on alchemy than on dynamics. None of us know this, which is a weaker part. Today we can say. But likewise, Darwin, natural selection of evolution, but he also gave a theory of pangenesis, which was very similar to Lamarck's. He didn't quote Lamarck as such, but he gave that theory, and which was wrong. And the worst is, Thomas and Morgan, whom we regard as father of modern genetics, did not believe in natural selection, did not believe in Mendel's laws, did not believe in chromosomal theory of inheritance when he started in 1900 his own career. Now these are something that we do not, and we need to know why they believe that, and why did this happen, and how did they change it. So in learning the process of science, we should examine how and why particular scientists held views that turn out to be wrong. Knowledge of historical controversies and misconceptions clarify the truly critical issues associated with a particular concept. Because progress in science is not a steady march to truth. It's not that it just goes on linearly. It follows a very tortuous path, back and forth and so on. And similarly, molecular biology is no exception. Even today, we have many misconceptions in its brief history itself. So what I plan to do is trace what things had happened. Now, how did molecular biology, the most exciting contemporary phase of everybody wants to do molecular biology today? And that's one problem. We forgot the classical conventional biology. And actually, as I often say, biotechnology, we learn neither biology nor technology. We just learn a few techniques. And that's where, partly because we do not teach in a holistic manner. But then, how did it originate? Goldschmidt Harvard Laboratory says molecular biology originated in the 1930s and 40s and picked up momentum in 50s and 60s. Although its direct predecessor was classical genetics, the emergence of molecular biology represented a convergence of work by geneticists, physicists, and structural chemists. That's true. That's where it has become truly interdisciplinary. And what it also says is, unlike biochemistry, which was already there in 1930s, people were talking about biochemical things and processes, which initially focused on proteins and enzymes, and only later turned to nucleic acids. Molecular biology's focus right from the beginning has been on gene, its various facets. But in the process, what has been ignored and forgotten often is that what we call as cytology, cytogenetics, embryology, and evolution, they had enormously contributed to this growth, which is not, often we talk of genetics, we talk of DNA, we talk of molecules and structure and so on, but how did we come to realize this enormous thing that happened in conventional, what we call cytology or cytogenetics, those techniques, and that's what I will try to highlight some of those things that happened. How did we come to this stage where Crick could propose the central dogma in 56? So one of the questions to go back in history, why did biology start? Main question, one of the questions, why life begets, why life? Why every animal, every plant produces its own kind? And that has been questioned. That's what led to the concept of biological heredity, that there is something that is inherited. How it is inherited, that was not clear, but historically, Hippocrates himself had suggested that mixing of semen from men and women in uterus forms embryo. But later on, his disciple, or in some way Aristotle, thought that sperm had nothing to do with it. It's only ovum had nothing to do, it only provides a ground on which the sperm makes the development of the new form. It was male-dominated society then, unfortunately continues in some way even now. But Anthony van Leeuwenhoek went step one further, who invented microscope. Under a microscope, when he looked at a human sperm, he drew a human form sitting inside the sperm. And for him, it was just a pre-formation. That sperm contained the entire body already, and it just grows. And that's why it looks like parents. So sperm and eggs were known by the 17th century, but their related contributions remained almost for the next 2 centuries unclear. Then people began to see the egg, drew under a microscope, and Schleiden and Schwann postulates that sperms and cells are ovum are also eggs and therefore equal. And this was extension of cell theory that they had given, which I'll talk about a little later. So by 18th, 19th century, the concept of that there must be some factors in these gametes which lead to the formation of next generation. And that's where the idea of inheritance and these things started. And Oscar Hertwig in 1876 actually discovered fusion of sperm and egg, nuclei using fertilizing sea urchin. That teaching sea urchin, you could see under microscope that here this is how the sperm enters and fertilizes. And mouse fertilization were drawn by Johannes Sobota, particularly taking the early stages, and beautiful drawings of that exist. So by that time, it was known that there is something that is passed on through these gametes. What was that? That was not clear. And so, but it provided a conceptual physical basis for biological inheritance. And then came cell theory, two great cytologists, which said cells is the unit of life, and then a pathologist by the name Virchow. I'm sure most of you would have heard his name, and he said every cell stems from another cell. This was an extension of theory, and more important was all diseases involve change in normal cells. This was his main hypothesis, and that's where, as a pathologist, he wanted to look at every time that what goes wrong in cells. Now, how did cell theory originate? That's again an interesting history. A dinner table discussion between two people. Schwann, an animal physiologist, and Schleiden, a botanist, both working under same scientists at Humboldt University. A remarkable example of transdisciplinary training. Today we are not allowed to guide a PhD in botany as well as in zoology, but that time it was, and this was the thing that led to them. And then they had a dinner in evening, and then Schleiden mentioned to Schwann about discovery of nucleus in a variety of plant cells by Scottish botanist Robert Brown. They were just discussing, and he said he has seen that he had talked about nucleus. Then Schwann remembers that he was looking at vertebrate notochord. He said, yes, he sees a similar structure in all those cells. And that led them to discussion and work. Finally, in Schleiden and Schwann independently published that cells and the nuclei are essential building blocks of plants and animals a year later. So they didn't publish a single paper, but because they worked together and because they started, so we talk of Schleiden and Schwann's theory. See, many of the theories, I'll come to one more example, which is not because they worked together, but because they worked, they interacted, or they may not even interact, but somebody thought that, yes, this is related, and that's how the name comes in. Now, something that Schwann wrote in his paper, important point was the elementary particles. So now you talk of particle, that cell is a particle, elementary particle of a body, are united by a common bond, the similarity of their formative principle that they give rise, one gives rise to another, that's how the whole body, multicellular body comes in. Notice the drawings, these are not photographs, they're all hand drawings. This is something I'll keep on reminding, that an art that we are really lost today, unfortunately. But then something more importantly, Schwann writes, which is true even today, that these and similar phenomena may remain for a time unexplained, although they merit the greatest attention and require further investigation. We will be allowed to leave them for a moment. Why? History shows that in laying down of every general principle, there are almost always anomalies at first, which are subsequently cleared up. Now this remains true even today. You come out with a new idea, there are criticisms, supports, and so on, and then things slowly resolve. See, why I'm quoting these as it is, it's not only we started thinking. The thought process have always been there. It's not that only with all the modern gadgets that we can think and talk about all this. The early breeding, we all have heard of Mendel, but hardly anybody would have heard of this Hungarian count, a very rich person who was a sheep breeder. And in 1819, before Mendel, he had given out these 4 laws. The last 2 and 3 are particularly same thing that Mendel also observed, that what is seen in grandparents can be seen in grandchildren, but not in sons and daughters. And the animals possessing desirable traits that have been inherited over many generations can sometimes have offspring with divergent traits. Some mutation, some gene combination that we often see that happens. So these were observed by him. But Mendel had been such a great name that we hardly know that what had happened before that. And of course, Mendel's things are remarkable. Particularly, he had no idea mitosis or meiosis, but he gave those pure conceptual things, and that's why we often think of genetics as a conceptual discipline. You have to imagine, and that imagination is what Mendel did, and he came out with these ideas. An important outcome and why Mendel becomes so important that he talked about particulate rather than blending inheritance, that there are specific factors, purity of gametes, purity of factor, that they remain, they don't mix up. And this was an important point at that time, because otherwise everybody thought that hey, nitrogen is blending. But he said no, they remain pure over the generations. But he was too far ahead of his time. Nobody appreciated Mendel. And actually Mendel died as a very sad person. He did write that I feel sad that nobody has recognized my findings. And when they were discovered, okay, before that, another development that happened was idea of chromatin. Today we are all excited about chromatin, but how did this term came in? Carl Nageli had seen cell division, but he considered an anomalous situation. He didn't realize that these cells are dividing. He has seen those structures. Walter Fleming, he used basic aniline dyes derived from coal tar. So they had to be chemist also. He found out his own dyes, stained structures, and saw thread-like structure in nucleus, which he named chromatin, because the colored threads, chromas, and he later on this chromatin was connected with word chromosome in the mitotic cells, and which Boveri, who had done a lot of work on this at that time, called them the chromatic elements. See, they had now we know chromosome and chromatin, but they had at one time in early 1900th century, chromosomes were also called spiremes because of the spiral coiling that one would see. And he published a book which has, this is fortunately available online, and I would urge you to see that book for the beauty of the drawings. For example, even this drawing of mitosis. Now, which part of structured cell you cannot see? He didn't have any confocal gland. He had a plain, simple microscope, and eyes and brain and analysis and good drawing. Now, how one would draw this? I don't know how many of you have seen this equipment. Camera lucida. Very simple. I myself have used it in my MSc and PhD in 60s because we didn't have cameras all the time, every time to take pictures and so on. It's a very simple device on the microscope. And this is the kind of microscope that he would have used. A simple ordinary microscope. On the top, on the eyepiece, you put something, a device which has a prism, so that one can see from top what is in the objective. But that prism also allows you to see what is on this piece of paper on the table. So you can look on microscope and with your hand keep on drawing what you are looking at it. And these were all drawn. I'm sure you can recognize and choose. He didn't know what these are, but he had drawn this. Even cellular components and everything is there. And this is what I think, when you look at modern gadgets, we don't see cells. We see only a marker, fluorescent color. And so we say this is tubulin, this is central, but we don't see the actual structure, the context where they are. And that's, I think, something that has, to me, an unfortunate situation in modern gadgets, that we don't see the whole thing. We see only a part that we want to see, like Arjun's pupil of the target only, nothing else. We don't need to be that. We need to focus on that, but also look at what the context in which it is. And actually, I have one slide where I put parallel confocal images of this, and I ask audience which one you like best. And without fail, it's these, not the confocal images, which provide the details. Anyway, now, so on one hand this had happened, that chromatin was known, and then in late 19th century, people were doing experiments, and three scientists who did it, the hybridization, De Vries, Correns, in 3 different countries, independently observed ratios in F2 hybrid progeny, similar to those published by Mendel. But then, as happens, as we happen now also, there was scooping attempts. Like De Vries did not want to cite Mendel. He knew what Mendel's were, but he did not want. Editor pointed out, or maybe reviewer pointed out, that there was somebody called Mendel who had done this, so he reluctantly acknowledged that, but he didn't want to honor Mendel as the father of genetics. He thought he was the one who had really put it together. Correns, who was also doing same time, learned about De Vries' work. The De Vries are trying to take priority, so he hurried his paper, got it published. Third person, Schermark, because they would have known that there are people working, so they sent their papers to him, and he had also obtained similar results. So he also hurried, sent copies of his paper, so that he can also claim to be a participant in rediscovery. Now this is very interesting. Now who will claim the priority? Who discovered the Mendel first? Theoretically Dave Rice. But then these two other people were clever, that they were more generous to Mendel than Dave Rice, suggested terms like Mendelism and Mendel's laws. And by saying that Mendel had already done this, they avoided the claim of priority. So now we say all three of them discovered it. But three never worked together. They had competition. And anyway, they discovered it. And soon after this was discovered, things started happening in both directions. One was Sutton-Bowery's chromosome theory of inheritance. I won't take much time, but I think each of you should read Sutton-Bowery's life. As a student, he was experimentalist. He even had a patent of cutting grasses mechanically, and that time he came across grasshoppers, which he gave to his teacher Macklin. And Macklin looked at its testes and saw beautiful meiotic chromosomes, which he ultimately studied, and that's where brought him to chromosomes. He became a surgeon and died because of a sense of duty. He himself was suffering acutely from acute appendicitis, but he did 4 emergency operations before he was himself operated, despite his pain. And 3 days later he died, very young. But that was a remarkable example, a bright person. And I'll briefly tell you how he came to this. And of course, Bovary was a remarkable person who has enormous contributions. And that's where Scott Gilbert in his book Developmental Biology writes about him, the last of the great observers and the first of great experimenters in biology. He had remarkable observations. And of course, I've seen these instruments that are lying in Würzburg University which Bovary used. Can you think that we can make such great discoveries with this kind of equipment? A very primitive microscope, and a dissection microscope, not even a compound microscope. Simple monocular magnifying lens. But these are the things that which he got this observation. And the experiment that he did was a natural experiment. In sea urchin, sometimes it happened that 2 sperms fertilize the same egg. He looked at them and looked at that the chromosomes segregate in very random ways. And that's what he indicated, that these are the possible ways. And then once you have multiple cells like this, you shake them and then let each individual egg develop to look at its potency. And even that only a few that developed as larvae, which didn't survive of course, but then only those who developed larvae which had at least one of each kind of chromosome. And that's where he came to the conclusion that each chromosome contains specific hereditary property. Oral chromatin quantity is not important. He never directly referred that this is what Mendel's factors are. But he did talk about that the inheritance factors are somewhere in the chromosomes. On the other hand, Sutton had really studied meiosis in grasshoppers, and that's what he writes. I may finally call attention to the probability that the association of paternal and maternal chromosomes in pairs and the subsequent separation during reducing division, as indicated above, may constitute the physical basis of Mendelian law of heredity. To this subject, I hope soon to return in another place. Everybody expected that he will do PhD, such a bright student, beautiful observations. But as I said, he went to medicine and chose a different period. And so that hope to return another place never happened. But the question is, Sutton and Bovary never worked together. Why do we call it Sutton-Bovary hypothesis? They never published together. They may not even have met actually. The credit goes to Edmund Beecher Wilson, a great embryologist and a cell biologist, or cytologist classically, but today we will call it cell biology, who authored the epic book Cell in Development and Inheritance. In the first edition, 1896 and 1926, I have seen this 1926 edition. It's a fat book. Beautiful drawings, of course, many the idea that he presents there, we don't agree, but then the analytical power was remarkable. And he was a key player in the chromosome theory of inheritance. Sutton's work was developed in his laboratory. He himself had worked with Bovary, with their good friends. See, in those days, American people go to Europe to learn science. Today, it has become reverse. But in 1890s, America was still kind of primitive. They will deemed it a great honor to be able to go to Europe and learn modern experimental biology, and Bovary's lab was one such place. Wilson publicized Sutton's research and associated to his friend Bovary's results, and he gave the term Sutton-Bovary chromosome hypothesis. So it was because of Wilson that we learned this hypothesis, although we never learned that these are independent discoveries. And Wilson and Native Maria Stevens independently established a relation between sex and chromosome, XX and XY and ZW chromosomes. That's when they connected that these are related to sex. That was one of the evidence that, okay, chromosomes carry factors of inheritance. But besides his personal contributions, he trained and influenced some of the young researchers, Alfred Sturtevant, Calvin Bridges, Herman J. Muller, who would collaborate with T.H. Morgan, his colleague at Columbia, in further substantiation of the chromosome theory of Mendelian inheritance. Now this again very interesting story. I'll come to this next time. Next slide. And one of his students, Marcella O'Grady, was teaching in a college in New York. He arranged her to go to Bovary's lab to learn, and she married Bovary, became his life partner. What is interesting again, unfortunate, is she worked in lab as much as Bovary did, but she was never an author. Even to get to join PhD program, a special permission has to obtain that a woman can join PhD. She was the first women scientist to join the PhD program under Bovary. After Bovary's death in 1915, she published much of Bovary's work, her husband's work. And there, maybe in some of those papers, her name comes in. Now, after this chromosome theory, the enormous upheaval, because no known evidence of an association of specific chromosomes to any observable characters. All that they have seen similarity. What Sutton had suggested that there are, unless you have all the chromosomes, you don't see the whole development. So the chromosomes must carry some information. And Sutton had seen that they pair and they separate like Mendel's independent assortment and segregation would expect them to happen. Cytological processes were not very clear. So the grasshopper one could see meiosis very clearly, but smoother organisms was not easy. And more important was, T.H. Morgan and Batterson, whose name has been very well known in genetics, were strongly opposing these ideas. The chromosome base of inheritance or thing. T.H. Morgan was really up in arms against this. It was only when Wilson's and Stevens' work showed that sex is a phenotype that it can be assumed sex chromosome, that some evidence that sex chromosomes carry genetic factors. Now, Morgan's is a remarkable case. Till 1910, Morgan vehemently opposed three of the most prominent ideas of the time. Darwinian natural selection based on small variations. He didn't believe in this. He thought what Debray said, clumps, sports or measured mutation, they are the only drivers of evolution, not what Darwin says are small variations. The newly discovered Mendelian genetics, he didn't believe in that there are factors that separate and so on and so forth, or that the genes determine early development. He didn't believe in that. And chromosome theory of heredity. And if you read Morgan's paper till 1910, you won't believe the same Morgan who was given Nobel Prize for discovery of the properties of gene. And who made him change? The students. Sturtevant, Muller, and Altenberg, and these students, when they discuss things, Morgan quickly reversed his ideas. Morgan went to develop a Nobel Prize winning vibrant research program that established modern genetics and paved the way for unification of genetics and Darwin evolution. So this is where it's important to learn what wrong ideas existed and how they corrected. The meanness as well as greatness, both one can see the human feature of this. Morgan's is a rather unusual case in which a scientist reverses ideas on not one, but three different theories in a relatively short period of time within 5 years. And all three, he completely reversed and became the champion of what he was opposing till 1910. And this is what he says in one of his papers in 1910, where he supports chromosomes are inherited now. Science advances by carefully weighing all of the evidence at her command. When a decision is not warranted by the facts, it is wise to suspend judgment until the evidence can be put to further test. This is the position we are in today concerning the interpretation of the mechanism that we found by means of which sex is determined. Now this is, I think, greatness of the science, of the scientist, who very modestly accept that where things were wrong. Now, when the gene, although the term gene didn't start in 1992 till it was given later on, but another thing that most of us do not know is of a medical person, Archibald Figueroa, who published a book in 1909, Inborn Errors of Metabolism, but had published his first paper in 1902 before chromosome theory was even published that way, where he showed that alkaptonuria is a rare recessive mutation in humans. So he was a remarkable doctor who will maintain entire pedigree history of each patient that he would see. Grandfather, father, children, wherever he could get that history, and based on that, and he would himself do the chemical analysis, why urine turns black, what happens. So he would do it, he had his own pathologist and chemist, and that's where he came out with this. And 1909, his book, Inborn Errors of Metabolism, is remarkable information. Then, of course, the other person was William Johansen, who gave the term genes, phenotype, and genotype, and gene as a unit of Mendelian inheritance. And the third person, of course, Thomas Malgova, who introduced Drosophila for genetic studies. That's another interesting history how we got to Drosophila. But I can't go on that because it will take a whole day to talk about all these stories. But they are very interesting stories. But during the next 2 or 3 decades, the fly room, that's where the Columbia lab was known, and it is still maintained as a museum piece. I'm glad that that remains as a pilgrim centers for anybody who is interested in genetics to visit fly room at Columbia. That unit of hereditary transmission, function, recombination material, all which he opposed till 1910. Then he became the most verbal and most authentic champion for these ideas. Then the other remarkable thing that happened was Sturtevant, as a 19-year undergraduate, laid the foundation of gene mapping. And this is where important some discussion happened. And this is exactly what Sturtevant himself has written. He has written a history of genetics, a kind of personal biography of how things develop. In 1909, Cassel published a diagram to show the interrelations of genes affecting the colors of rabbits. Cassel was working on the pigment genes in rabbits, how the gene interaction, these things happen. It seems possible now that these diagrams were intended to represent developmental interaction, but they interpreted that they represent how genes are located, one gene after the other, and so on and so forth. And so that's what he writes. But they were taken at Columbia as an attempt to show the special relations in the nucleus. In the later part of 1911, in conversation with Morgan about this attempt, which we agreed had nothing in its favor, I suddenly realized that the variations in strength of linkage already attributed by Morgan to differences in the spatial separation of genes offered the possibility of the determining sequences in the linear dimension of genes. And so I went home and spent most of the night with the neglect of my undergraduate homework. Like most of the students, the semester class test and presentation, all these pressures are there. But that's what he says, neglected this, worked whole night, how to put these in order, and that ultimately developed the gene maps over the next 10 years. And this was minded an undergraduate. We just joined lab and discussion. And this is Morgan's lab. When he moved out of Columbia in the University of Texas in Austin, and that's we say during scorching Texas summers, that's as hot as North India is. Initially, Muller set his bottles of flies in pans of water draped in wet cheesecloth to keep them cool. I can relate it because sometimes, I know 20 years ago, we had a major power failure on our campus. Maybe 30 years ago. No power for 5-6 days. How do we maintain our Drosophila? That's what we did. That we put a big earthen pot, put all the vials in that, kept it wet, and kept the fan on. I took them home because home we had electricity. We kept the flies alive. So that I can see what was happening. What is important is then later on his head gave him an air, so-called what we call air condition. This is the original Patterson unit that he had, the Sturtevant unit. I don't know why it's called Sturtevant name, whether it was Sturtevant geneticists who designed it or some other company, but that's what it was called, a big unit that will keep the lab cool. The other interesting thing to notice is in this figure, this is Muller standing. We don't examine flies this way, but that was the way that they would do. Use a jeweler's loupe, a magnifying lens fixed on the eyes to see the flies standing. And that's what Muller would be doing. And in this lab, besides being a place for many original concepts, Muller's Nobel Prize winning discovery of radiation-induced mutations was made in this lab. That's where he discovered the CLB stop and the X-rays-induced mutations, and that's the story. The other young person, Calvin Bridges, again, most people do not know about him. He really revolutionized genetics as we learn today. Has a very short life, hired by Morgan in 1911 as a lab helper to clean the fruit fly bottles, and later he was allowed to pursue independent research. Now, what was his major discoveries? Primary non-disjunction, which a rare event. He saw one fly, but he was convinced that this fly is not a contamination. He pursued and discovered non-disjunction, which led to the launch of the journal Genetics, Genic balance theory of sex determination. He was father of modern genomics in a way, because he mapped genes on chromosomes, physical mapping. We today talk of human chromosome physical mapping started in late 70s, not true. Physical mapping was done in 1930s into Drosophila chromosomes. Using cytogenetic methods, you have deletion, okay, that mutation mutant allele is missing, so therefore that gene must locate in this chromosome, and he had made detailed maps of that. His repeat hypothesis, when he looked at chromosomes, he saw certain polytene bands to be repeated, and he suggested these may be evolution related. And that's what led E.B. Lewis to work on Bythrox locus and win Nobel Prize. So it was, and this is what E.B. Lewis himself has written about how Bridges' repeat hypothesis helped him to do all that study that he did. Now, other important things that Bridges invented was introduced dissecting microscopes, ether anesthesia that we all use today. It was Bridges who discovered, and you can see in this picture, although stereobaric was treating, but Muller is still using his old habit of looking at flies. But that's what Bridges changed slowly. He introduced incubators, where you can maintain temperature, and he introduced the food that we use today. And as a communicator, he would share flies across the world. It was his responsibility in Muller's lab to do that. Introduced Drosophila Information Survey, DIS, which you have some of them in collection. So this is the interesting thing. 1933, this was the first of a kind of preprint service. Whatever published in DIS, Drosophila Information Service, was just for communication between fly workers across the world. And as you submit a manuscript, type, it's retyped and published as it is. No peer review, no corrections, nothing. And I have been following the DIS because till 1990, till we had internet access and so on, that was the only source available every year to know what's happening in Drosophila across the world. And I don't remember reading any paper that was published, note that was published in DIS, which turned out to be wrong. So there was a real preprint survey that happened and had helped Drosophilists across the world. It was started by Bridges. And he would collect notes on every mutation that he had. He built a totem port, 4 sites. Each gene that is known, put a note of it on each face of that thing. And that's what is used after he died in 1939 by his student Bremen. And that's what we call Bridges and Bremen. That was a Bible in that till 1992 when Litz and Grell's genetic variations came out. And now, of course, we have Flybase, which has taken up all this. But these are good beginnings to be understood that how systematic work can help maintain, develop things. So fly room, as Morgan describes, I received, this is when he wrote a memoir for Bridges in 1940. I received a grant from Carnegie Institution of Washington, Career and Heredity. Bridges and Strutman were appointed to undertake independent research. They were already working in his lab, Bridges as a lab assistant kind of thing. They were appointed to it. And then, notice something, the bananas hanging. That was the fly food. And that's what Morgan describes. These were the days when bananas were used as fly food, and in one corner of the room, a bunch of the bananas was generally on hand. And adjunct to our researchers, which interested other members of the laboratory in a different way. You can pluck a banana and go away. So and that's what is recorded that, yeah, this was a common thing that would happen. And as there were no incubators, a bookcase and a wall case were rigged up with electric button, bulb, and a cheap thermostat. How many of us can think of that situation. That's where the great genetics foundations were laid, which behaved badly at times. Milk bottles came to the program at an early date, but where they came from was not known. I don't know how many of you have seen old milk bottles, glass milk bottles. None of the generation would have seen it. That's how today we get in polypack, but earlier it used to come in glass bottles, which had to be returned next day. So somebody would pass them to the fly lab. It was only after some time, as Morgan writes, at a later date we were bold enough to ask for a case of new bottles. And that the last part is important. This was the condition not intended to suggest that we were handicapped. On the contrary, our proximity to each other led to cooperation, everything that went on. So this is a record. In Morgan's lab, this is what Alderman had recorded. Nobody knows whose idea came in from. Somebody was author, and they were all individual authors. Morgan never put his name in the publication with students because he believed that unless he had contributed with his own hands to a discovery, he won't be an author. And this was another thing that happened in Muller's lab. Polytene chromosomes were identified as polytene, and this is the paper that was first published. And the title of the paper is Salivary Chromosomes and the Attack on the Gene. And this is the concluding part that Painter writes. With these four discoveries before us, that is constant and distinctive patterns, somatic synapses, the behavior of active and inactive regions minded. At that time, nothing was known about what genes are, how they are, and what, but he did talk about the behavior of active and inactive genes and the separation of the arms of the large autosomes. It was clear that we had within our grasp the material which everyone had been dreaming. We found ourselves out of the woods and upon a plainly marked highway with bypaths stretching in every direction. It was clear that the highway led to the layer of the gene. Now this is remarkably true, and that has actually happened. What he said was actually polytene chromosomes have contributed enormously to the way we understand gene action, gene regulation, and so on, which is unfortunately not recognized so much. Another thing that happened was Bridges prepared these maps, looking at chromosome, camera stood a drawing, and he mapped each, drew it as a line, and he drew 5,000 such lines on the entire polytene nucleus. And this was made over several years. 1939 he died, his son P.N. Bridges took it over and completed the maps. So, and these are now standard reference maps. In 1960, people thought, okay, with EM we can see more maps. So they would take squash preparations, cut thin sections, and look in here. And multiple images, they couldn't find a single extra map. And they couldn't find that there was something that Bridges had drawn is not absent. Now this is really remarkable, that Bridges' power of observation, precision, and perseverance. Mind it, no fancy microscopes, ordinary monocular microscope. That's what was used, and till date this remains our standard reference for each gene that we know. And what Bridges had done was map different genes on each of these maps, wherever it was known, and that's where the physical mapping started. So in 1930s, where we come now to the state is, gene is the unit of function, unit of mutation, and unit of recombination. But Muller in 1922 itself has raised, Muller was a remarkable thinker. He had great ideas. In 1922, he had raised the possibility, it will even be possible to determine whether the entire gene changes at once, whether when we say unit of mutation, does the whole gene change in one go, or its parts can change? Whether gene consists of several molecules or particles, one of which may change at a time. Muller had raised this possibility, which was ultimately similar. Benzer in 1950s worked on R2 locus, where he cistron, neutron, recon, the fine structure of gene. Incidentally, Benzer was a physicist, worked on transistors as part of our effort so that the radars can function better. But listen to Ernst Schrödinger's What is Life, Delbrück's lectures, and he knew about Morgan's genetics work. So after PhD, he jumped to genetics and thought Drosophila not a good, started using bacteriophages, R2 locus. After finishing that, he jumped to Drosophila now. He is father of chronobiology, father of behavioral genetics, father of neurobiology. All the the Drosophila models of human degenerative diseases in the 90s were generated by Benzer. A remarkable person. The other important thing that happened, which I'm sure you all know, Barbara McNichol discovery of jumping genes. against the established idea that each gene has a defined locus. And Barbara McClintock, who had contributed to that gene mapping in maize and tomatoes, now suddenly comes with the idea that some genes don't have defined position. And so she says that from examination of instability of genic action at a number of known loci in maize, it is concluded that mutations need not express changes in genes, but may be the result of changes affecting the control of genic action. Now mind it, by this time when these statements are made, if nothing is known, what is gene? What's the physical chemical basis? But they had these conceptual ideas, and which is what we need to appreciate. The other thing that has not completely forgotten is the subtle description of heterochromaty, which was discovered by a German scientist, Emile Heiss, working with bryophytes. He looked at the whole mitosis process directly on microscope, sitting for 24 hours. Just how does the cell go through mitosis? And then he noticed certain regions remain condensed all through, and that's what we call heterochromatic, differential chromatic. Heteropyknosis, differentially stained, or differentially condensed chromatic. And genetic studies in Drosophila soon suggested that heterochromatic regions do not carry Mendelian genes or typical genes that were being mapped. And other evidence came in that heterochromatic is inactive, inert, and gene desert, and so on till 1960s, but later on things changed. But what is important for molecular biology to remember is the whole idea of chromatin biology, chromatin modification that we talk today so much of histone or DNA, that started with searching what is heterochromatic, what is condensed, what kind of changes happen. So the first experiments on methylation, cytosine methylation or histone methylation, was to understand this cytological entity called heterochromatic. Then in 1930, a new branch happened, that's what we call cytogenetics. I still call myself a cytogeneticist. I don't want to be ashamed of this primitive, what is considered primitive. I think it's very important. Firstly, it happened because of correlated with chiasma and homologous chromosomes that are cytologically seen, and genetic recombination. And that's why it's cytogenetic, that you look at both the processes. Physical maps of genes on polytrin chromosomes that I talked about briefly, and then chromosome rearrangements and heterochromatic, that many times in different populations, there are chromosome arrangements involving heterochromatic. So how heterochromatic influences what happens, that's where cytogenetics branch happened, and it did bring chromosome in focus. Genes were working in, but chromosome in focus was because of this branch cytogenetics. And interestingly, X inactivation that we today talk about, this gene was not discovered to begin with. X inactivation was proposed by Mary Lyon purely in cytogenetic studies. Likewise, hyperactivation of X chromosome in Drosophila, the ROX genes, that came much later. It was pure cytology, morphology with X chromosome, and cytogenetic studies that my supervisor Mukherjee had done in Biermann's lab to show that X chromosome is hyperactive, and to which I also contributed my PhD in 1970s. So these are pure chromosome studies that happened. And what polytrin chromosome studies did, most people do not realize that all our understanding of gene action, that chromatin opens, started with these polytrin chromosomes. In 1950s and 60s, when they looked at these chromosomes at different developmental stages, they could see that same region undergoes dramatic change in morphology. This is condensed here, slowly opens up, and goes back as development proceeds. And this was as a result of ecdysone stimulation. So that's where it was known that hormone action, particularly steroids, activate on certain set of genes, which then activate other sets of gene, cascade of gene activity. So this was the beginning of our understanding of hormone action and molecular biology of hormone. And of course, that chromatin opens up during activity. So based on these observations, the whole field of stress biology, heat shock proteins and heat shock genes, were discovered in Drosophila, a serendipitous discovery by Franco Retosa, who is known for his work on ribosomal DNA more, but he was the one who found the polytrin chromosomes, and subsequent studies on metabolic label proteins revealed the heat shock proteins in Drosophila, and that's where the whole field of stress biology started. And so the molecular biology of stress responses beginning in cytogenetic studies in Drosophila. And of course, my own contribution in a way, discovery of first non-coding gene in Drosophila. Because polytrin chromosomes were there, we could see that, okay, when you treat these cells with amides, a specific gene is activated. And that we showed in 1982 is a non-coding gene, which is again very rarely recognized, that yet this was one of the first evidence that a non-coding gene exists and is important. Now, I come to briefly, maybe can I take 10, 15 minutes more? It's taking, okay, how embryology changed in developmental biology. Morgan, who was opposed to chromosome theory, did believe that pre-formation is not the case, that what Leeuwenhoek had suggested, but there's a kind of epigenetic development, that something happens the way the things happen in embryo. Of course, for him at that time, epigenetic was not gene changes. Epigenetic was something that happens after what is inherited. And he talked about that. And so that linked epigenesis to heredity. And now the question was where these factors are located in the embryo, what time they act, how do they act. This became a hot question. And two people who worked on this happened to be disciples of same teacher for their PhD, Wilson and Morgan. They were colleagues at Columbia, but they had very bitter arguments because Wilson thought nucleus is the seat where these factors are located. Morgan, on the other hand, said nucleus under the is called cytoplasm in embryonic development. Cytoplasm determines how the cells will move, nuclei will move, and so on and so forth. And they're bitter arguments. And they could be sometimes really bitter, but they were good friends. And like in other cases, Morgan reversed his views. By 1910 or 15, he had agreed that just its nucleus is more important. And then in 1930, there was a real tussle between embryologists and geneticists. Embryologists, a great name like Frank Lilly, Hans Spemann, who got Nobel Prize for the discovery of organizer, Harrison, Just, they all were against genetics. Because, like even today happens, that we think this field is more important than other. Geneticists believed embryologists to be old-fashioned, and that development would be explained through gene expression. That's what geneticists thought. Embryologists regarded geneticists to be uninformed about organism development and felt genetics to be irrelevant. And this is what one quote from Harrison in 1937, who warned his fellow embryologists, beware of wanderlust of geneticists. And the summary of this is that genetics has nothing to do with embryo development. Let's be careful because it can block embryology studies. Now this is typical that happens. But those days scientists would write things very clearly and without mincing words. It was C.H. Weddington, a geneticist, an embryologist, and evolutionary biologist, who synthesized the whole thing. And he said the relationship between inducer and competent tissues parallel that of genes and cytoplasm. Now, he combines both Wilson's and Morgan's strands, that your genes as well as cytoplasm. And neither cytoplasm nor nucleus can be disregarded. In fact, the most important subject to discuss is how they affect each other. And he emphasized epigenetic process through canalization, that each cell modifies its gene activity and thereby moves to a different direction, determination, and that lies at the root of cell differentiation. Now, it's often a very interesting question. Morgan, an embryologist, by training an embryologist, started using Drosophila, but never studied in embryology. Partly because its embryo development is very different from any other sea urchin or a frog or mouse or anything. So they thought it's very bizarre and it's not worthy of being studied. And that's what Morgan did do it. But interestingly, these same unusual properties have today become real prize for development biologists. They have become most important tools to do this. And one of this was reflected in this Nobel Prize discovery of Lewis and Nusslein Follard, and that's where I wrote a small commentary that the Nobel Prize is a tribute to the power of genetics. That pure genetic studies, we could come to them. And then, of course, things have changed since then. Combination of formal genetic analysis with contemporary cell and molecular biology. Now today we know how evolution has shaped these Hox genes and developmental genes, but the roots were in very interesting ways connected with what happens in embryology, with what happens in gene action, and so on and so forth. Now, I'd like to quote something interesting thing. Bower in 1904 had talked about how to understand chemistry of chromatin. Now, notice this. For it is not cell nuclei, not even individual chromosomes, but certain parts of certain chromosomes from certain cells that must be isolated and collected in enormous quantities for analysis. that would be the precondition for placing the chemist in such a position as would allow him to analyze the hereditary material more minutely than the morphologist. For the morphology of the nucleus has reference to the very least of the gearing of the clock, but at best the chemistry of the nucleus refers only to the metal from which the gears are formed. Gears, I don't know, in a clock. How many of you would notice that in old clock there were very minute gears and which had been crafted very well. That's what talks about, that the whole process is because of these sequential activities and process that goes on. Now, what he talks that we will need enormous amount of this. That's what molecular biology helped, articles that we now don't need, but still those who do chemistochromatine have to talk about millions of cells to isolate a nuclear or this fraction, that fraction. Muller had also predicted molecular biology in 1922 in a very different context. De Herelle, a French scientist, has found that in dysentery bacilli, there causes production of loose motions, and in that liquid, what he didn't know but must be bacteriophages that kill these bacilli. So he noticed this. And when Muller noticed that okay, there was something in that liquid that would kill the bacteria specifically, he made this statement that two distinct kinds of substances, the de Herelle substances and the genes which they work with in Drosophila, should both possess this most remarkable property of heritable variation or mutability, which working by a totally different mechanism is quite conceivable. It would open up the possibility of two totally different kinds of life working by different mechanisms. But then he said more importantly, if these de Herelle bodies were really genes, fundamentally like our chromosome genes, they would give us an utterly new angle from which to attack the gene problem. They are filterable, to some extent isolable, can be handled in test tubes, and their properties as shown by the effects on bacteria can then be studied after treatment. It would be very rash to call these bodies genes, and yet at present we must confess that there is no distinction between the genes and them. Hence we cannot categorically deny that perhaps we may be able to grind genes in a mortar and cook them in a baker after all. That's what molecular biology says, cooking a gene or cooking something or something. And the other thing is, must we geneticists become bacteriologists, physiological chemists, and physicists? Mind it, this is what molecular biology became ultimately. But he had predicted this with being zoologists and botanists, let us hope so. Unfortunately, today we become physical chemists, physicists, bacteriologists, but not biologists, not zoologists or botanists. And that's where I think something is missing in our learning process. Now, early insights, I quickly go through. I said Garrod had already talked about it. And then came a classical experience of Efrusi and Biddle and Efrusi, who transplanted imaginary discs that will make eyes later on between different genotypes to find out how each gene controlled to the pigment formation. And the method that they used, as he said, transplanted a mutant eye imaginary disc into wild-type larvae, demonstrated distinct diffusible substances manufactured by wild-type alleles of the vermilion cinema loci, which control distinct steps in the formation of brown pigment. So now we knew that how to gene action. Each produces something, they interact and they produce the final pigment that we see. These studies require genetically defined stocks, while the techniques employed were those of experimental embryology and biochemistry. And so this did not affect the gene themselves, but the way the product will interact and develop. And this led ultimately to Biddle tattoo neurospore experiments, which defined one gene, one enzyme happens. So before we knew the genes, we knew that genes are there somewhere on the chromosomes, and they contribute to some enzymes. And then, of course, the other thing that we do not know is something Kaspersson, a great cell biologist in Sweden, who in 1930s had designed a cytospectrophotometer. How to measure by light absorption and thereby deduce the amount of DNA or RNA and so on. I had the fortune of meeting him. Actually, he had visited Varanasi, and I had pleasure of sitting by him being my pillion rider. That's remarkable, thinking that those crowded roads. But what was already shown was constant diploid chromosome numbers, that every species has defined chromosome number, what we call 2-1 or diploid number, and gametes have haploid number. Constants of DNA, 1C and 2C values, gametes have 1C, fertilized egg had 2C. This was all established by cytology and cytospectrophotometry. And this is what led to Avery et al.'s demonstration in bacteria to be applicable to eukaryotes as well. that yes, because DNA only is the constant material in these, not RNA or protein. That was one important point that led to DNA being established as genetic material. And of course, cytology, he made another remark that DNA is a polymer. Till that time, I thought it's only 10 monomers that combine to make nucleic acid. But they showed it was a polymer by themselves. And so cyto and histochromic studies between 1940 and 1950 also brought RNA in focus. Because what they could show by staining of different cell types, that cells that are active in secretion have more RNA than cells which are inactive. And that brought in RNA somewhere in between DNA and protein. And that's what was instrumental in Crick's central dogma, that RNA occupied the central space. And of course, what Crick had suggested in central dogma, this is his hand drawing in 1956 presentation, which finally published was 1970 as central dogma of molecular biology. But the prototype was here. And what he said was only information flow, that this cannot happen. I'm sorry. That this reverse flow, once information go to protein, it cannot flow back to RNA or DNA. This is what central dogma was about. But the misinterpretation was RNA cannot have independent function. This happened because of a simple sentence that Watson used in his molecular biology of gene, first edition: DNA makes RNA, makes protein. And then people thought RNA only does make protein, nothing else. And that's what led to the whole idea that we culminated in selfish and junk DNA. That non-coding DNA, which doesn't make a protein, is useless. And this was outcome of the belief that functionless DNA persists selfishly. This is what they call the selfish, that once it gets into the genome, we can't get rid of it. Now this is something was fundamentally wrong. And because this was out of ignorance, since if you go back in history, way back in 1890s, Bovary had drawn this beautiful picture and described phenomenon of chromatin diminution. That this cell will become precursor somatic cell, this will become the germ cell precursor. Somatic cell loses most of the chromatin, and only from this cell, all the somatic cells of the worm will arise. Germ cell all through retains the entire chromatin. And this is what he said, chromatin diminution. So what we know now is, germ cells know how to get rid of, the genome knows how to get rid of something that we do not need, but it's retaining germ cells because it has essential function. What Crick and others talked about selfish DNA, they didn't know about this history. That heterochromatin or condensed chromatin or non-coding DNA, repetitive DNA, satellite DNA, they all are very important for germ cell function, for evolution, for new genes and so on. Because they didn't know this, even in ciliate macronucleus, 95% of DNA present in the small micronucleus is lost. And micronucleus is equivalent to germ cell of eukaryotes in ciliates. And the macronucleus, the large nucleus, is somatic equivalent. So genomes indeed know how to get rid of silence or silence heterochromatin or selfish DNA when not needed. And now, of course, we know they are master regulators. I have written many reviews, and there are hundreds of such reviews and thousands of articles every year. We talk about non-coding RNA. Actually, non-coding RNA today have become the fashion. What were to be completely ignored 20 years ago, now they are the one thing. Now the question is, unlike Morgan, why did molecular biologists not realize the fallacy of selfish or junk DNA for nearly 2 decades? This is something that historians can ask. I raised this question in one of my reviews, that it would be an interesting issue for the historians and social scientists to find reasons as to why the misinterpretation of central dogma and the concepts of selfish junk DNA continue to hold sway for decades. My suggestion was, perhaps the linear reductionist approach and the race to publish and find applications prevalent in recent times were some of the contributory factors. We need to publish, and we become fashionable, we follow fashion. Because if you, actually I know that working on non-coding RNA as I have been, in India I could get funds, but in Europe and US, they wouldn't get funding. Even great scientists, if they want to work on non-coding RNA in 90s, sorry, no funds. Why waste money on selfish DNA? This is what unfortunately happens. And so a better understanding of biology and history would have helped molecular biologists to avoid such reductions and influences. And I would urge everyone to think in a holistic manner, not in a reducing manner, because the philosophy that E.B. Wilson has stated in 1915 was Science should teach us to keep an open mind, to look facts straight in the face. It should help to deliver us from the deadly vice of thinking we know things of which we are really ignorant. It should lead us to place a higher valuation on observation experiment than on authority and precedent. Not merely in the spirit of sportsmanship does science play her game. She also strives to realize an ideal, one that is very plain and simple, and this ideal is, in a single word, progress. That's what science looks at. Not to solve the ultimate riddles of the universe, if such there be, not to attain to absolute truth, but to advance knowledge. Such is the aim of science. And I think if we remember this, our subsequent history will be much different. So with that, I'm sorry I took long than what you expected.
Thank you very much. We have a little time for questions. We've
Okay,
got 15 minutes for questions. And there are some questions online as well. So if anyone with questions, feel free to put up your hand. No? Well, while this takes a little time, I'll read out a question that we had.
Well, questions are very important. That's what Wilson also says, and everybody says that science progress only when we question, when we ask. And we ask difficult questions. We ask something which may appear very naive to begin with, one may be embarrassed. Should I ask this question? But unless we ask, we do not clarify our own thinking. And that's why I urge everyone to ask more and more questions. In any case, I'll be around here for 7 days. Anytime, any of you, if you want to talk, want to discuss or ask, I'll be available. I'm doing with the Drosophila training workshop, so I'll be here for next 7 days. So you can take longer time to think, but if you have some questions right now, please go ahead.
I'll read out one question that came in online. What we know about Drosophila-based genetics research is mostly from Morgan's work, but is there anybody who worked on Drosophila genetics before Morgan?
No. Drosophila was not work for genetics anywhere. It was look for taxonomy and so on, but not for genetics. It was Morgan, and this again interesting because this question is asked. I'll just go a little into history. That he had visited an entomologist in US lab who were working on taxonomy of Drosophila. I forgot the name, the scientist. So he gave him a bottle of Drosophila melanogaster. At that time it was called Drosophila amphiphila, not melanogaster. That okay, you use this. This is white Drosophila, harmless, very quick to grow, easy to grow in lab. Within 2 weeks you get next progeny and large numbers, so good for genetics. And that's how Morgan brought it. And 1909 he got the first white eye mutation. During the early period, they put Drosophila to all kinds of torture. High-speed centrifugation, high temperature exposure, mechanical shaking, whatever they could to get variations, get mutation. And what's remarkable is, by 1913, between 4 years, they had 72 mutations that they had mapped. Now this is remarkable, and that's where Drosophila kept on growing very fast. So, but before Morgan, no one did Drosophila for genetic studies. Yes.
I think I can ask.
Yeah.
So I think Crick's diagram is the same, where it says what may happen or what may not happen, like what can never happen. It's interesting that he says protein cannot form protein, protein cannot form RNA, protein cannot form DNA, but he doesn't say RNA cannot form.
Well, he could say dash line.
Yeah, but I mean.
RNA, that's again very interesting. The discoverers of reverse transcriptase did not get Nobel Prize till Watson made that a solid line. So it's again interesting. Watson believed that reverse transcriptase is only viral property, RNA viruses. And so people who claim that they see reverse transcriptase activity in human cells in culture, he said there must be some virus lurking behind. So till he was convinced that yes, reverse transcriptase actually happens, check and others, then only they could get Nobel Prize. It's a historical fact. That what Crick didn't initially believe. So only later on that he agreed that okay, this can also happen. That's what it is. See, history, if we read it, we get to know many, many interesting things. And I really enjoy learning these things because they give us a sense of our own context. How and where did we ask? And I can share that my PhD work went on very smoothly because I did read history of the problem that I was working on, of what are the ideas. And that's where I knew, even I realized Muller's idea was wrong. You see, in the 60s, to think that Muller, the great man, had a wrong impression, I believed it, did experiments, and yes, there was something to be differently thought of. So don't ever be burdened by, because some great name has given this, how can you go against it? Like my own paper, 1980, everybody talking about selfish DNA. And then I discovered a gene which is not making a protein. It should be selfish. But I never believed it. I believed that it must have a function and continued. And fortunately, it was only because I was working in India I could get funds. And partly what you said, Bhatnagar Prize, part of that was because of my work on this gene. So I suggest that in India sometimes things can be very different than what is in US or Europe. Here we can, okay, money is less, but sometimes we can ask questions that we want to ask. And that's where I've been very happy about it.
So what functions are now attributed to these so-called selfish genes?
What functions are not attributed, except for making protein? Say, I give a simple analogy. This institution, research institution, there are research students, there are faculty. How many non-research, non-academic community? Almost equal number, maybe somewhat less. What are they doing?
Facilitating learning this.
Exactly. Regulating, facilitating, deciding when to do what. We are sitting here, he didn't go run around and find out everyone. Office did it. That's regulation, and that's what non-coding things do. Besides making proteins, ribosome RNA, tRNA, they actually do, but others are deciding when, where, how much, and so on. That's what the regulatory part.
And what made you think these selfish genes must have some function?
Well, because believe in evolution, that natural selection is not helpless, that you can't get rid of something which is not doing anything. Because imagine energetically, every base to be added to a DNA requires a triphosphate. Now, cell will spend billions and billions of triphosphates every time it replicates for doing nothing. I don't think natural selection of living system is so weak, so helpless as selfish DNA concept made it to be. It's not. And of course, I knew about this old classical work, that chromatin diminution is not something that cannot happen. Because I learned about it partly because I was teaching, and so I read, and that's the great benefit of teaching, that you read about something, and then you know, and then you begin to connect it. And so I was convinced. I know sometimes when I wrote, I had very strong personal comments on blogs. Again, because I was counter-arguing against selfish DNA. There were strong comments. I said, nothing doing it. And I feel this, I've been right, not utterly wrong.
So basically, it's a sort of philosophical outlook derived from natural selection.
Well,
Maybe
partly because you know biological system heterogeneity is there. Heterogeneity is known to play an important role in evolution. Because, say, repeated DNA, satellite DNA, they are present everywhere. But then what is interesting is, two related species, sequences are completely different. Selfish DNA say, okay, this because there is selfish, there is no selection. My argument and others who argued about it is, these small subtle differences decide whether this regulation will work at this time or at another time. And that's what it turns out to be now. All right, thank you.
Thank you. Thank you, Dr. Lakhotia. Thanks everyone for being here. And,